Welcome back to Biology 1406. Today we're going to be talking about membrane transport, which is basically how things get from the outside of the cell into the cell, or how products that are made inside of the cell and waste products that are made inside of the cell are removed from the cell. The first thing that we need to talk about when we think about membrane transport is what the membrane is that we're working with. And we're talking about the plasma membrane that surrounds the cell. If you remember, we talked about that as a phospholipid bilayer, which is that dual layer of phospholipids arranged tail to tail, with the heads that are hydrophilic, water-loving molecules, and the tails that are hydrophobic or water-fearing molecules. This membrane is a fluid structure. It's not a solid structure. It's not like a plastic bag around something. It is really mobile and sort of works like a waterbed. So if you push one side of a waterbed, the whole bed will ripple, but nothing will actually leak out of the waterbed, theoretically. Embedded in that membrane, we have some lipids and some proteins that are able to sort of move around within that membrane as they need to. And we have some cholesterol in there, which serves as a stabilizer. It keeps that membrane fairly structured and makes it so that it's not quite so fluid that it just falls apart. It is a barrier. It separates the inside materials of the cell from the extracellular fluid. But in order for a functioning cell to survive, we have to be able to get nutrients in. Oxygen, carbon dioxide has to get out. Other products and wastes, so proteins that we make through the endomembrane system that we discussed when we talked about cell structure. Those things have to be removed from the cell. Waste products, like I said, carbon dioxide, has to leave the cell. And so we have to be able to get things back and forth across that membrane. We use the term permeability to describe how we move things into and out of a cell and how easy it is for something to move into and out of a cell. If it is a structure that you physically cannot get anything through, for instance, a wall, we refer to that as being completely impermeable. If, on the other hand, it's more like an open doorway, that is something that we refer to as being freely permeable. The intermediate in between that is what we refer to as selectively permeable or semi-permeable. And I usually describe that as a screen in a window or a screen door. That screen allows some materials, some molecules to come in and out fairly easily. It allows oxygen in, it allows carbon dioxide out. It allows some smells and those sort of chemical odors in and out of the house. But it stops larger molecules. It stops insects. It stops larger molecules from being able to get in very easily. The ability of molecules to travel through that selectively permeable membrane, which is what our plasma membrane or our cell membrane functions as, is really based on molecule size, the electrical charge of the molecule, the shape of the molecule, and the lipid solubility. When you go through some of the mastering assignments, you'll notice that there are some questions in there about size and lipid solubility. And it makes the most sense when you think about it that something that is lipid soluble would be soluble in a fat, would move fairly easily through a phospholipid bilayer, phospholipids being part of that group of biological molecules that include the fats. Smaller molecules obviously are going to move easier through the membrane. And then when we talk about electrical charge and molecular shape, we're going to talk about variations depending on what membrane you're working with and what kinds of channels and gates you have available. So outside of the cell, we have this matrix of materials. It's a variety of different structures and objects and molecules that are surrounding and attached to the outside of the cell. I'm not going to ask you to know details about these. You need to know that they exist. You'll get more details about these things when you take cell molec as you move forward in your degree. But you do need to know that the extracellular matrix is primarily made up of glycoproteins, collagen, proteoglycans, and fibronectin, and that embedded inside of the phospholipid bilayer, we have these proteins, these molecules called integrins. And we're going to talk a little more in detail about how those work with the cell to help move structures into and out of the cell. So when we start talking about moving things across the membrane, we have to start talking first about what is going to drive the movement across the membrane, which direction the molecules are going to go, and why. And that is generally based on a concentration gradient. Concentration gradients are based on the molecules that you'll find on either side of a substance. So if, for instance, I have a plasma membrane, and on one side of the molecule, or one side of the membrane, I have all of these, we'll call these sodium molecules, and inside of the membrane, I only have a few sodium molecules. What I have is a really high concentration of molecules outside of the membrane and a lower concentration of molecules inside of the membrane, and giving me a concentration gradient from high to low that flows in this general direction. Because these are sodium ions, they're also positively charged anions, giving me also an electrical gradient because I have a higher charge, a more positive charge on the outside of the membrane than I do on the inside of the membrane. And together, when you combine both the actual concentration of the molecules themselves along with the charge gradient that comes with those ions, we get what we refer to as the electrochemical gradient. That electrochemical gradient will drive transport across the membrane. Depending on which direction we're following that gradient, we will either be using a passive process, which moves in a direction from high to low, or we will be using an active process, which moves in the opposite direction from low concentration to high concentration. And when we talk about passive processes, we talk about three different processes. We're going to talk about all three of them here in a minute. The first is simple diffusion, the second is facilitated diffusion, and the third is a very special case of diffusion called osmosis. Active transport is going to include both generalized active transport and vesicular transport, and we'll talk about those details here in a few minutes. So we're going to start out talking about passive processes. Diffusion is the simplest of the forms of membrane transport that we're going to talk about, and is really the simply the movement of molecules from a high concentration to a low concentration across a semipermeable membrane. One of the things you need to remember here is that all molecules are constantly moving, and that movement is a completely random pattern, so that as molecules are sort of colliding with each other randomly in the air around you, and they are bouncing off of each other, moving in opposite directions, we will get this sort of random mixing of solutions. So if you, for instance, take a black cup of coffee and pour some cream in it and just let it sit on the counter without actually stirring it, over time the cream molecules in that coffee will actually diffuse throughout the entire cup, so that eventually you will have a completely mixed cup of coffee. This is sort of the pure definition of diffusion in action. We're talking about the concentration gradient here, and in passive transport, the most important thing that you need to remember is that the molecules are always moving from an area of high concentration to an area of low concentration, and that they do so randomly and without any energy output by the cell. That movement is influenced by a few things. The first of those things is the steepness of the concentration gradient. So if you look at this figure here, at the very beginning of this experiment, we have a really high concentration of our dye at the bottom of this flask, and at the top we have a really low concentration, and so our concentration gradient is going to move from the bottom of the flask to the top, and this is a really steep gradient because there is no dye at all at the top of this molecule, at the top of this flask. The temperature will also influence how fast these molecules move. Molecules will always move faster in a warmer temperature. The mass of the diffusion substance, the smaller the molecule, the faster it's going to move because it's going to be less likely to interact with any other molecules. The surface area of the molecule, is it a nice, smooth, round molecule, as round as that is, or is it something that maybe has some more structure to it, or it's more likely to interact with other molecules, get spun off in another direction, those sorts of things. And then, of course, there's the distance between the high concentration gradient and low concentration gradient. How far does it have to go to diffuse? In this particular flask, the distance is actually fairly short, but if this were a taller flask, you would have a significantly farther distance to travel, and therefore you would have a longer diffusion time. So when we talk about diffusion, we're going to talk about two main kinds of diffusion. We're going to talk about simple diffusion, and we're going to talk about facilitated diffusion. Simple diffusion is just simply the movement of molecules across the membrane from an area of high concentration to an area of low concentration. The molecules that are going to move easiest through this membrane are going to be those molecules that dissolve in fats, lipid-soluble molecules like alcohols and steroids and fatty acids, as well as some dissolved gases like oxygen and carbon dioxide. They're literally just going to move directly through that membrane without any assistance, with no energy expense, moving from the area of highest concentration to the area of lowest concentration. When we talk about facilitated diffusion, we're still talking about moving from a high concentration to a low concentration, but instead of moving directly through the membrane, we're going to use either a channel or some kind of carrier. Now, channel-mediated diffusion is all about charge and molecule size. When we talk about opening a channel, we have to have the right charge set up in place for that to happen. Carriers, on the other hand, are entirely based on the molecule that's being moved. So if you take a look at this figure here, what you'll see is that glucose fits perfectly into the little active site in this protein, and the minute that glucose is in that protein, it allows that protein to change shape and move that glucose molecule from the outside of the cell to the inside of the cell. So when I talk about carrier-mediated transport, the first thing we need to talk about are the details about the carriers themselves. And these carriers have three very specific characteristics that you need to know about. The first is specificity, and what that means is that one transport protein will only transport one particular kind of molecule. A glucose transporter won't move calcium, for instance. It will only move glucose. Saturation limits. These function a little bit like what we talked about with enzymes, where It doesn't really depend on how much of a particular compound you have to move as much as it does how many transporters you have to move them. So for instance, if you went to a concert, it doesn't matter how many of you are outside of the concert hall. What determines how fast you get in is how many doors they have open and how many people they have working the doors to check tickets. Regulation is the third characteristic you need to know about. And this is just the fact that there are certain hormones and other molecules that will control when these transporters will do their job. For instance, glucose molecules will only be transported into a cell after their transporters have been triggered by insulin. Insulin is released from the pancreas when blood sugar levels are high. It flows over the outside of the cells and indicates to the glucose molecules that blood sugar is high and there's glucose to be moved. And then the transporters then get to work and do their job. Now, transport happens with carriers in one of two ways. The first of these is co-transport. And with co-transport, I am going to move my molecules two at a time in the same direction. And if I don't have both molecules in my carrier at the same time, then I'm not going to be able to move them at all. Sort of like if you've ever been to an amusement park and you go to get on a ride and they put a stranger in the seat next to you because they have to have two butts in every seat in order for the ride to go. Counter transport, on the other hand, is the idea that we're going to move one substance into the cell at the same time as we're moving a substance out of the cell. I always like to use the example of IHOP on Sunday morning for breakfast. You go, you wait in the lobby, and as soon as somebody leaves, then you can go in. But you cannot go in until somebody has finished and left. And so that is counter transport. So we're going to talk about tonicity, which relates back to osmosis because we're talking about the movement of water, but we're talking about how the movement of water will affect the shape of a cell by comparing the solution outside of the cell or the extracellular fluid with the solution inside of the cell. So we're comparing the solution that is external or the extracellular fluid to the intracellular fluid. In these three very special cases down here, the first of those solutions that we're going to talk about is an isotonic solution. And in this particular scenario, we have an equal concentration of water on both sides of the cell membrane. So the intracellular fluid and the extracellular fluid have an equal concentration of water. And because of that, water is going to move into and out of the cell at about the same pace. And when it does that, the cell is basically going to stay about the same shape and about the same size. It's not really going to affect the size or the shape of the cell much. In a hypotonic solution, we have an external solution or an extracellular solution that actually has a really high water concentration and an intracellular solution that has a relatively low water concentration. And because of that, water is naturally going to want to go down its concentration gradient from high to low and move into the cell. Now, in an animal cell or in a cell in your body, if water moves into the cell in an unchecked or uncontrolled manner, it can lead to something called hemolysis of the red blood cells or lysis of general cells, where the cell will eventually become so full it will literally pop like a water balloon. And we refer to that as hemolysis or lysis, depending on what kind of cell we're talking about. In a hypertonic solution, we're talking about a situation where we have a low concentration of water outside of the cell and a high concentration of water inside of the cell. And that is going to lead to a situation where water is going to want to flow in the direction of its concentration gradient from a high concentration to a low concentration. And it's going to leave the cell, causing the cell to shrink or crenate as a result of that concentration gradient. We're going to move on now to the active processes, and the big difference between the active processes and the passive processes is the requirement for ATP or energy of some source to be available to actually make these processes work. Passive processes don't require energy at all. So if we go back to the bicycle example, a passive process would be sort of coasting downhill on your bicycle where you're not having to do any work or exert any effort, whereas an active process would be how much energy you exert turning around and coming back up that same hill. So in an active process, we're going to move in the opposite direction, and we're going to move from a low concentration to a high concentration. And that's going to cost us some energy because we're going upstream or uphill. Okay, so primary active transport versus secondary active transport. This is a really complex topic that we are not going to spend a ton of time on in this class. This is something that you will get in more detail. You'll get a little bit of secondary active transport when we talk about cellular respiration, but for the most part, this is stuff that will come up in more detail in a cell molet class. The only thing you need to know, the literal only thing you need to know about primary active transport versus secondary active transport is the energy source. So in primary active transport, I get my energy directly from ATP. In secondary active transport, I get my energy from a stored energy that comes from either a hydrogen or a sodium concentration gradient that we use to drive another substance, to create something else. You'll actually see the use of a proton gradient or a hydrogen gradient when we talk about cellular respiration to drive the process of ATP. So if you can just remember that primary active transport uses energy directly from available ATP, whereas secondary active transport uses energy that is stored in the form of a concentration gradient of hydrogen or sodium, then you'll be good to go with this topic. Vesicular transport is another form of active transport, and in this case you need to remember back to when we talked about vesicles during the cell structure lecture. And what we're going to do here is we're going to move products in or out. This is bulk transport of materials, where we're moving large materials into or out of the cells, and we're going to use vesicles, or those transport mechanisms that we talked about, to do that. If we want to move stuff into the cell, we're going to use a process called endocytosis. And endocytosis comes in three forms. It comes in the form of receptor-mediated endocytosis, it comes in the form of pinocytosis, and it comes in the form of phagocytosis. So we have receptor-mediated endocytosis here. We have phagocytosis here. And we have pinocytosis here, so that you can see what's happening with the differences between them. So with receptor-mediated endocytosis, what you want to pay attention to are these receptors that are on the outside of the cell membrane. And the way that this works is that molecules will come and they will bind to these receptors, and these receptors will then be folded into the cell into a vesicle, and they will end up inside of that vesicle with the molecules that were attached to them on the outside. And eventually a lysosome will come along, or an endosome of some sort will come along and break this down and release those products to be used. But what you need to remember is that the receptor-mediated endocytosis leads to the products binding to the receptors on the outside of the cell, and then those products and receptors end up inside of the vesicle, inside of the cell. With phagocytosis, we've talked about this a little bit. This is when the cell actually reaches out parts of its cytoplasm in the form of a pseudopod, that's that word right there, and wraps itself around a product on the outside of the cell. Now, this is a somewhat more specific process than pinocytosis. Phagocytosis is going to reach out and grab a very specific molecule and pull it into the cell in the form of a vesicle. Pinocytosis, on the other hand, works more like a straw. I described in class pinocytosis like if you were chewing gum and you blew one of those backwards bubbles where you put the gum in front of your teeth and sort of sucked the bubble into your mouth. That's the same sort of thing that the cell is doing here, where it's pulling in part of its membrane, and along with that membrane comes whatever happens to be in the vicinity of that part of the membrane when it pulls it in there. And so this is a very less specific mechanism of moving things into the cell. It just sort of gets whatever is in the extracellular fluid there. So all of these are methods of getting materials into the cell via endocytosis. This process is how we both get things out of the cell in terms of exocytosis and how we get to deeper layers of cells via transcytosis. So with exocytosis, we're simply going to create a vesicle from the plasma membrane and release those contents out in the extracellular fluid in a vesicle. You can sort of see, or I'm sorry, outside of the vesicle. So you can see here in this image, a vesicle moves close to the membrane, and as it becomes a part of the membrane, the materials inside of that vesicle are then ejected from the cell. Cellular wastes can be used here, proteins that need to be exported from the cell and sent off into the body. There are a lot of different things that can be that exocytosis can be used for. Transcytosis is sort of a unique combination of both endocytosis and exocytosis. And this is a situation that allows materials to get through multiple layers of cells to get to a particular cell that it needs to get to. So for instance, if material was coming in from here and it needed to get to here, it would have to undergo endocytosis to get into this cell, and then it would have to undergo exocytosis to get out of that cell, and then endocytosis again to get back into the target cell. Because we're doing both endocytosis and exocytosis and just simply moving through the cell, we call that transcytosis.